Blasticidin S HCl is a chemical compound that is widely used in molecular biology and biotechnology research. It is an antibiotic derived from the bacterium Streptomyces griseochromogenes and is commonly used as a selective agent for the maintenance and selection of cells that have been genetically modified with blasticidin resistance genes. Blasticidin S HCl acts by inhibiting protein synthesis in both prokaryotic and eukaryotic cells. It specifically targets the peptidyl transferase activity of the ribosome, preventing the formation of peptide bonds during translation. This leads to the accumulation of incomplete and non-functional proteins, ultimately causing cell death. One of the key applications of Blasticidin S HCl is in the generation and maintenance of stable cell lines expressing exogenous genes. Researchers often introduce blasticidin resistance genes into cells alongside their gene of interest. These resistance genes encode enzymes that can inactivate or modify blasticidin, rendering cells resistant to its inhibitory effects. By adding blasticidin to the culture medium, only cells that have successfully integrated the resistance gene and the desired gene of interest will survive and proliferate. This allows for the selection and enrichment of cells that have been successfully genetically modified. Blasticidin S HCl is also used in the study of gene function and protein localization. By introducing blasticidin resistance genes into cells alongside genes of interest, researchers can study the effects of gene knockdown or knockout on cellular processes. This is often achieved by using techniques such as RNA interference or CRISPR-Cas9 gene editing to specifically target and disrupt the expression of a particular gene. The addition of blasticidin then allows for the selection and isolation of cells in which the targeted gene has been effectively silenced or knocked out. Furthermore, blasticidin S HCl is commonly utilized in the production of recombinant proteins. By incorporating blasticidin resistance genes into expression vectors, researchers can select and amplify cells that have successfully integrated and expressed the desired recombinant protein. This is particularly useful in the production of therapeutic proteins, enzymes, or other biotechnological products. It is important to note that blasticidin S HCl should be handled with care, as it can be toxic to cells and can have adverse effects on human health if mishandled. Proper safety precautions should be followed when working with this compound, including the use of personal protective equipment and adherence to appropriate handling and disposal protocols. In summary, Blasticidin S HCl is a valuable tool in molecular biology and biotechnology research. Its ability to selectively kill cells lacking blasticidin resistance genes allows for the maintenance and selection of genetically modified cells, making it an essential component in the generation and study of stable cell lines expressing exogenous genes.